భరత్ సిస్టమ్ లోంచి కూడా లాగిన్ అవుతాను. సిస్టమ్ లోంచి లాగిన్ అవుతాను. హలో, నీ వాయిస్ వినిపించట్లేదు. హలో, వాయిస్ వినిపించట్లేదు నీది.
Hello. ఏ మనకి మీ ఇష్టం హాఫ్ అన్ అవర్ 10 minutes 20 minutes మీ ఇష్టం. Right. చెప్పండి.
Am I audible?
Yes, ma'am.
Okay, can we start the session?
Yes, ma'am. We can start.
Okay.
Yes, ma'am.
So good afternoon, one and all. In continuation to the Laboratory Professional Week, day second, we are going to have a topic, and today's speaker is Bharat Kumar Reddy, and he is a STA from Biochemistry Department, Super Specialty Hospital, and his topic is extracting DNA. Okay, over to Bharat. Please continue.
Yes, ma'am. Yes, ma'am. Good afternoon, all. Today we are celebrating National Medical Laboratory Professional Week. On continuation of this professional week, we are conducting webinars. Today we are taking one more webinar. First of all, I say that happy National Medical Laboratory Professionals Week to all the medical professionals.
Any difficulty, Bharat?
No, madam. 1 second. Today I am. My name is Bharat Kumar Reddy, working as a STA lab in ESA Super Specialty Hospital, Sanatnagar. Today we are going to discuss a very important topic, that is DNA extraction from blood by using a particular kit what we are using in our hospital, that is QIAGEN kit. So actually, what is the basic source of DNA? So where we can get DNA? The routine is blood. We can collect our DNA from blood. Other than blood, so many other substances we can collect and extract DNA. These include seminal fluid, and then urine, saliva, hair, and then swabs, teeth, and bone, and feces, fingernails, and bite marks. And then another important is tissue. From tissue also we can extract DNA, like formalin fixed tissue also and paraffin embedded tissue. So from extraction from DNA from tissue is very important for molecular pathology studies. Here some of there we are using for forensic laboratories like hair and swabs, and then bite marks. These are used for forensic studies. So these are all the sources of DNA where we can get DNA. And then, so what are the essential components of DNA extraction? So what we extract and what is why we are extracting DNA. So for this components include maximize DNA recovery, and then remove all inhibitors. So we can extract DNA means we have to remove all inhibitors, and then maximize the quality of DNA. Sometimes we can extract DNA, but the quality is not up to the mark. So that is also we cannot use for diagnostic purposes. And then remove or inhibit nucleases. So these nucleases are a particular enzymes that which cleaves the DNA or digest the DNA. If the DNA extract having all these nucleases, it causes continuous digestion of DNA, and we can lose the DNA native structure. And then sometimes we can extract either double stranded DNA or single stranded DNA. So some of the viruses we can we can extract single stranded DNAs. And then, so what is the quality of DNA may recover? For example, how much DNA we can recover from blood, or how much DNA we can recover from seminal fluid. So all these points also very important. So how much quantity we can extract from different sources. For example, a diploid cell contains approximately 6 picograms of DNA. And then the average WBC of an adult is 5 to 10 and into 10 to the power of 6 cells per ml of blood. The theoretical recovery of DNA per microliter of blood is 30 to 60 nanograms. And then semen contains approximately 3 picograms of DNA. So based upon our quantity, we can extract from different sources. So majority we can extract DNA from WBC. So for each microliter of blood, we can extract up to 30 to 60 nanograms of DNA. So this is a good sources of extraction of DNA in our diagnostic purposes. And then how much DNA we need? For example, we can extract 10 nanograms of DNA. Then we can say that, yes, this is sufficient for us. But what is our need? How much DNA we have to recover? We need. For example, for the PCR reactions, an average 1 nanogram of DNA is sufficient. Why? Because for the PCR reactions, we can amplify number of copies by doing this PCR. So very less quantity of DNA is sufficient for PCR reactions. For example, we are doing RFLPs, that is restriction fragment length polymorphism. These procedures, we need high concentration of DNA. For example, it requires 50 nanograms of DNA, it is required. So based upon this, for the PCR reactions, it's very, very minimal amount of DNA is sufficient. Why? Because we can enlarge, we can double the number of DNAs by using this polymerase chain reaction. For theoretically, many of the commercially available kits are very sensitive to extract 100 to 250 picograms of DNA. So it is very, very less quantity also we can extract from the available kits. Coming to the next slide. So how many types of extraction is there? So there are so many types of DNA extraction are there. For example, these are the types, various types of DNA extraction. The first one is organic extraction. So here on organic extraction, the phenol chloroform we can use. And then non-organic extractions. Here, proteinase K and salting out method we can use. So these are all the different methods of extraction. And the third one is Helix, means it is a ion exchange resins are there. These ion exchange resins are bind this DNA and elute after elution. And then one more important method is that is FTA paper method. FTA means Flendel's Technology Associates, means it is a cotton-based cellulose paper containing chemicals that brush the cell, denature the protein, and protects DNA, leaving a sample suitable for molecular identification without the risk of disease contamination. So this is the paper like this. It's just like our filter paper. It's having round circles. We can take drops of blood in that circles. So that cotton-based cellulose paper contain all the digestive enzymes are there. So it digests all the cells and denatures the proteins and it protects the DNA. So whenever we want DNA, we can just punch some part of this filter paper. Again, we can extract the DNA. And the final one is silica-based extraction method. Here it is having a silica exchange resin. This resin will binds the DNA. Whenever we elute the adding the elution buffer, the DNA will elute it. So these are all the types of DNA extraction methods. So now we are using in our laboratory, that is QAM DNA mini and blood kit. Here these kits are commercially available. It is very suitable for normal conditions. It does not require so many sophisticated equipment. So very minimal equipment, we can extract a good quality and quantity of DNA from this kit. Okay, introduction of the QAM kit. So these QAM DNA mini kit provide fast and easy method for purification of total DNA, reliable for PCR and short-run batting. So this is the very minimal procedure are there. It does not require so many sophisticated instruments. It does not require any refrigerated centrifuge like that. It requires very minimal equipment. And then the total DNA, that is genomic, viral and mitochondrial, can be purified from either whole blood, plasma, serum, buffy coat, bone marrow, other body fluids, lymphocytes, culture cells, tissue and forensic specimens. So by using this kit, we can extract DNA from many sources, but each sources it require a lysing method. So the lysing method is different from each substance. The remaining part, that is after lysing the substance, the elution of DNA is common. The only thing is, based upon the substance, we can use different techniques for lysing of that sample. And then purification requires no phenol and chloroform extraction or alcohol precipitation and involves very little handling. These are very advantage. So it does not require so many biosafety cabinets. Does not require anything. We are having very minimal equipment and pipettes. We can extract this DNA in high concentration and good quality. And then the purified DNA is free of proteins and then nucleases and other contaminants or inhibitors. So this is very important. So we can extract a good quality of DNA, but we are having so many inhibitors and proteins, it causes denaturation of DNA. Okay. So what is the principle of this extraction by Poisson method? The principle is very simple. First, we have to lysis the cell by protease or proteinase K. This is the first step. The lysis of cell is very important. And then purification on mini spin columns by using adsorption to the membrane. So these columns having a special type of membranes. These membranes are catch the DNA and it will release all the unwanted material. Whenever removal of residual contents, that is removal of all digested material, proteins, all are removed. And then the final step is elution of pure nucleic acids. So this is very simple principle. First, we have to lyse the cells by protease or protein K, and then purification is by using adsorption of membrane, removal of residual contents, that is unwanted things we have to remove, and final step is elution of pure nucleic acid. This is the simple principle of DNA extraction. Okay, the important step in DNA extraction is, that is lysis, that is importance of proteinase K. So this proteinase K is used during DNA extraction to digest many contaminating proteins present. So this proteinase K is used for the digestion of all proteins like cell membrane, nuclear membrane, everything it will digest. And then it will also digest nucleases. So these nucleases are inhibitors of this DNA. These nucleases will digest the DNA. That's why the removal of nucleases is very, very important. And then what is the mechanism of this proteinase K? This proteinase K cleaves peptide bonds next to the carboxyl group of N-substituted hydrophilic, aliphatic, and aromatic amino acids. It also cleaves peptide amides. And this proteinase K is most active at temperatures 50 to 65 degrees centigrade. And coming to the equipment and reagents. So these equipments are reagents and equipments are, we require 96 to 100% of ethanol, and then 1.5 ml microcentrifuge tubes, and then pipette tips with aerosol barriers, and then microcentrifuge, or it is also called as mini spin or microcentrifuge. See here, it does not require any refrigerated centrifuge. So all these procedures we can done at 15 to 20 degrees centigrade, that is room temperature. And then we require vortex for mixing, and then we require water bath. So is water bath or heating blocks. These heating blocks are very suitable for DNA extraction. And phosphate buffered saline. So it is required for dilution of samples. Okay. So what are the preparation of reagents? So this kit contains proteinase stock solution. It is stored at 2 to 8 degrees centigrade. Pipette 1.2 ml of protease solvent. It is given one solvent and one powder form of proteinase K. Just we mix these 1.2 ml of solvent into this powder lyophilized form. Then it can store at 2 to 8 degrees centigrade for 12 months. So it's very, very simple procedure. And then we required buffer AL. So buffer AL means it's a lysing buffer. So mix a buffer AL thoroughly by shaking before use. So it is ready to use. And then this lysing buffer is stored at 15 to 25 degrees centigrade. And then buffer AW1. So buffer AW1 means it's a washing buffer. So we have to wash the unwanted substances. For that, we require washing buffer. So it is also mix a 50 ml of 96 to 100% of ethanol we can add in that bottle. So the A1W buffer is ready. And all these are stored at room temperature. Okay. Then buffer AW, that is wash buffer 2. This wash buffer also same for removal of unwanted substances. We can use this wash buffer. So directly add 50 ml of ethanol, that is 96% to 100%, and then it is also stored at room temperature. And final, that is buffer AE. AE means it's a elution buffer. So after removal of all the unwanted substances, the DNA is bind at the membranes. So when we are adding this elution buffer, along with this elution buffer, the DNA will extract. And then it requires QAMP mini spin columns and then collection tubes. So these are all the reagents we can use for extraction of DNA. Coming to the sample material. So we are saying that we are extract DNA from blood samples. So suitably, EDTA sample is suitable. Along with EDTA, we can use heparin and sodium citrate also. But the best thing is we can use EDTA specimen. So we require, how much quantity is required means it required 200 microliters of whole blood. For example, if we can already say that the WBC count is very low, then the extraction of DNA is also very low. For that reasons, some of the specimens we require Buffy coat. So whenever the leukocytes and these things are very less, then we prefer for Buffy coat. So normal count of WBC we can use blood is the good method. And then tissue. So it requires 20 milligrams of tissue it requires. And then if we are extracting from diploid cells, it requires 5 into 10 to the power of 6 of cells. So these are the sample material. For example, suitably we can use EDTA samples. Once we collect the EDTA sample, the best method is we can extract within few hours. Otherwise, these samples can be stored 2 to 8 degrees centigrade up to 1 week. If we have more time means we can store at minus 80 degrees centigrade. So all these are suitable. Next, coming to the actual protocol. What is the protocol? Step-by-step procedure in extracting DNA. This is a very, very, very simple method. We have to take micro cuvettes, that is microcentrifuge tubes, and then we can pipette 20 microliters of proteinase solution. This proteinase solution is required for digestion of all protein material. And then add 20 microliters of 200 microliters of sample. We can take a microcentrifuge tube, add 20 microliters of proteinase, and then add 200 microliters of sample. For example, sometimes the sample is very less. For example, if the sample is only 100 microliters we are get, then the remaining 100 microliters we can make up with phosphate buffer saline. We can't add 100 microliters directly. So we can dilute these 100 microliters with PSB buffer to make 200 microliters and add 200 microliters as a sample. This is the first step. Then add 200 microliters of lysing buffer and then mix thoroughly by vortexing. So we can add proteinase, 200 microliters of sample and 200 microliters of lysing buffer. Mix thoroughly by vortexing and then incubate at 56 degrees centigrade for 10 minutes. So what is required for this incubation? So the proteinase K is having highest activity at 56 degrees centigrade. So that's why at that temperature we can incubate for 10 minutes. Okay. Then next step, add 200 microliters of ethanol. Mix thoroughly by vortex mixing and briefly centrifuge the tube to remove the drops from the lid. Okay. And next step, here it is very important. Here we are introducing these mini columns. So this small centrifuge tube is there now. It is a mini spin column. It is having the silica gel membrane. This green one is a silica gel membrane. And this empty one is the collection tubes. We can place this in this collection tube and we can centrifuge. So actually we are preparing lysing in the another centrifuge tube. Now transferred all these into this mini spin column. Okay, and place this mini spin column into a collection tube, and then we can close it and centrifuge at 8000 rpm for 1 minute. So after centrifugation, what will happen? The unwanted material we are added, the material we are added in the spin column. After spin, the unwanted material will collect in the collection tube. So we remove this collection tube and place this column in another new collection tube. It is clear? Okay. Here, place the QAMP mini skin column in a 2 ml collection tube and add 500 microliters of washing buffer. Okay, and centrifuge 8000 rpm for 1 minute. Okay, and then place the QAMP mini spin column in a new 2 ml collection tube and add 500 microliters of washing buffer 2. Okay, so washing buffer 1, it removes some of the unwanted substances, and then we can one time we are removing the unwanted material by washing buffer. Here the centrifugation speed is 14,000 rpm for 3 minutes. Okay, and then discard the flow through and collection tube, and then place the QAMP mini spin column in a new 1.5 ml microcentrifuge tube and add 200 microliters of elution buffer. So this is the final step. We are adding the elution buffer or distilled water. So distilled water means it is a nuclease RNase free water we require. Otherwise, we can add elution buffer and incubate this at 1 minute at room temperature and centrifuge 8000 rpm for 1 minute to elute the DNA. So this is the summary of procedure. First, we are adding 20 microliters of proteinase K and then 200 microliters of sample and 200 microliters of lysine solution. Okay, we can vortex it and we can incubate 10 minutes at 50 degrees 6 centigrade. Okay, so after the completion of incubation, so this is the substance. We can add 200 microliters of ethanol and mix by vortexing. Then all these material we transferred into a mini spin, and then we can kept in a collection tube and centrifuge. Okay, first we can remove all the unwanted material and we are adding washing buffer 1 and then centrifuge and remove the unwanted substances and transfer this mini column into a new collection tube and add washing buffer 2 and then transfer this to a new collection tube and centrifuge at 14,000 rpm for 3 minutes, and you can remove this collection tube. Now it is having a purified DNA which is attached in the columns, and then transfer this into a new collection tube and add elution buffer. Then centrifuge it, you can get a pure DNA sample. So this is very simple, and it required hardly 20 to 30 minutes of time. Okay. Now we are getting a DNA, but we don't know it is a good quality. So how much purity is there, how much yield is there, and how much length of DNA we can collect it. So this is very important, just like a quality check. We can check before we can proceed for PCR and another application. If we are not check this quality, if we don't know it is having a high amount or low amount or high amount of another substances are there or not. For this, we require a nano drop. It is a instrument. It's a nano drop instrument. Nano drop instrument is nothing as a spectrophotometer. But spectrophotometer, we require high quantity of substances. For example, the light path of this spectrophotometer is 1 centimeter. That is, it requires 450 to 1 ml of solution is required. But we are getting only 200 to 250 microliters of DNA from the extraction. So we can't measure by using the spectrophotometer. That's why they introduce a simple spectrophotometer called as nano drop. Why it is called as nano drop means it requires a 1 microliter of substance. Once again, I am telling it is required only 1 microliter of sample. That's why it is called as nano drop. So what we are measuring in the nano drop? The purity is determined by absorbency at 260 nanometers and 280 nanometers. And then the purity check is based upon the 260 and 280 ratio. So this is the graph we are getting from nano drop. Here we are measuring the substances at 260 nanometers and then 280 nanometers. And we can take the ratio of these two, that is 260 by 280. This ratio is 1.7 to 1.9 means the quality of DNA is very good. And then this nano drop will give the graph, means you see here the peak. This peak should be the quality of this DNA. And then it will give the concentration also, like 45 milligrams like this. The main important is the 260 by 280 ratio is in between 1.7 to 1.9 means we are extracting a good quality of DNA. Okay. So coming to the next, what about the length? So quality is good. What about the length? So most of the PCR reactions, it does not require, it does not necessary to measure the length. Why? Because in the PCR, we are amplifying the DNA based on what we target. So we can use the target DNA, we can multiply so many times. That's why the length is a not issue. But in some of the DNA methods like RFLP, we require how much length we are getting. For this, the DNA should be concentrated by alcohol. For this reason, this pulsed field gel electrophoresis is the method to get the length of the DNA. This pulsed field gel electrophoresis is just like electrophoresis, but here it is pulsed field. Pulsed means every 30 to 40 seconds, we can change the interval, switch intervals. That's why it is called as pulsed field gel electrophoresis. So for this pulsed field gel electrophoresis, it required TE buffer, that is tris EDTA buffer. So this EDTA buffer is mostly used for the storage of this DNA sample and mixing of sample, dilution, for mixing of dilution. It having the pH of 8.0. And then, so avoid drying the DNA pellet for more than 10 minutes at room temperature. And then 15 to 25 degrees centigrade, since over dried genomic DNA is very difficult to dissolve. And then load 3 to 5 micrograms of DNA per well and started pulsed field gel electrophoresis condition as follows. Here, what is the agarose concentration? So the 1% agarose gel is we are using, and then 0.5x TBE buffer, that is tris borate EDTA buffer is we are using. And then what is the switch intervals? Switch intervals are 5 to 40 seconds. And the runtime is 17 hours. And the voltage is 170 volts. So little bit you are confusing about this pulsed field gel electrophoresis. In this picture, you have clearly understood what is meant by routine electrophoresis and pulsed field electrophoresis. Here you can see the gel here. This is the gel, agarose gel. And these are the electric fields. You see here, 4 electric fields are there. Anode, cathode, and anode, cathode. So routine electrophoresis, we can run one end to another end. But here, every interval, we can change the direction at 120-degree radius. First, it will run like this. After 60 seconds, it will run like this. So that's why it is called as pulsed field gel electrophoresis. So every 40 seconds, the direction of this is changed. Okay. So how to see this? So this is the gel. We are adding sample 1, sample 2, sample 3, and one marker. So after staining with ETBr, ETBr means ethidium bromide. So after staining with ethidium bromide, it will show like this. So all these DNA bands are there. So we are using this marker. This marker is just like a standard. So it is having 230 kilo base pairs, 315, 577, 817 like this. So this marker having the base pair length of 230 to 1500 kb. Based upon this marker, we can compare our samples. For example, here in sample 1, we are having 2 bands. One is at approximately 1300. Another one is approximately 700. Like that, we can measure the length of DNA. If more length is there, it will run very slowly. If slow with very less amount of length is there, it run very fastly. 230 kilo base pair means it is a very less DNA. That's why it fast very far in the electric field. So based upon this, we can say that the length of our extracted DNA. Okay. So these are the instruments we are using to identify the bands of this DNA. This is called as gel documentation system. So gel documentation system is used for the measuring of these bands. So it shows bands like this. So these are all the samples 1, 2, 3, 4, 5, and these are all the length of base pairs. So these are the original pictures of gel documentation system. See here, it is clearly mentioned that this is a marker. First one is the marker, and then remaining all are the samples. So based upon the extraction, the length of DNA we can yield. You can see here in second sample we can yield the maximum of 500 length of base pair length of DNA. Some of sample number 10 we can extract 631 kb length of DNA. So based upon this we can measure the length of DNA. Here the faster the run, the length is low. The slower the run, the length of the DNA is high. So this is the very simple method to identify the length of our extracted DNA. Okay, this is in brief about the extraction and quality and quantity of DNA. Thank you very much.
Thank you, Bharat, for an elaborative talk. I was thinking whether I'm attending a CME or a workshop. So each and every step you have explained it very detailed, and it was very informative. So any doubt from the audience, they can always ask.
Yes, ma'am.
Any questions? One question from my side. What are the clinical conditions in which you do this test at our laboratory setup?
Madam, actually, we are doing pharmacogenetics, madam. Tacrolimus, if the drug is suitable or not, so many variants are there. We can use this pharmacogenetics. We can increase the drug, madam, levels, tacrolimus drug levels.
Okay. Thank you, Bharat. Yeah. Any questions from the audience? Yes, Jayachan, you wanted to ask something?
Yes, Jaychan.
Okay. Thank you, Bharat, for your extensive talk. Hope everybody enjoyed today's session on extraction of DNA and its application. We'll meet up again tomorrow at the same time with another talk. Thank you one and all for participating.
Thank you, ma'am. Thank you for your cooperation and support, madam. Thank you.
Thank you one and all. Thank you, Bharat.
Thank you.